Hi everyone, welcome to Carl Chem's Journal Club. I'm Lady, and I'm an analytical biochemist with a master's in chemistry and 10 years experience. This video is for you if you want to learn something new about recent advancements in biochem, and you won't need an advanced degree to understand the video. Today I'm going to talk about the article shown here. HIV-1 capsids enter the FG phase of nuclear pores like a transport receptor, and I'm able to share it here with you because it's licensed under Creative Commons. If you want to look it up yourself, the DOI link is in the description, and you'll be able to access the entire article for free. This article proposes a new way HIV-1 can potentially enter the nucleus of a cell. The authors are claiming that the HIV-1 capsid can enter the nucleus of a cell through nuclear pore complexes, or NPCs. Nuclear pore complexes, as their name suggests, are pores in the nuclear membrane that allows cargo to enter and exit the nucleus of a cell. One of the first things I like to do when I read an article is look at the journal it was published in and the academic institutions that the research came out of. So in this case, we are published in Nature, which is super high impact journal, and we're coming out of Max Planck and MIT. I really do have high expectations for this paper coming out of the gate, so let's get started. First, I'm going to go over some background to provide some context. The picture you're seeing is from Figure 6. It shows the HIV genetic material in purple, which is inside the capsid, which is blue and gray. Before the HIV enters the cell, the capsid is actually enclosed by another membrane called an envelope, which isn't shown because the paper is really focusing on the capsid here. So we already know how HIV enters the cell, where the envelope will bind to CD4 receptors on CD4 positive T cells. It'll fuse with the host cell membrane and then deliver the capsid directly into the cytoplasm of the cell. This paper focuses on the HIV capsid and how it gets the HIV genetic material into the host cell nucleus. So I'm really not going to spend any more time talking about how the HIV works in its cycle and how it gets into the cell. I'm just going to focus on the capsid itself. The capsid is made out of many copies of the same small proteins, which are pictured in blue. These proteins assemble together like Legos to form a structure that's about 120 nanometers in length by 60 nanometers in width. So it's about twice as tall as it is wide. For the HIV infection to be successful, the viral genetic material must enter the nucleus of the host cell. We really aren't 100% sure how the HIV gets through the nuclear membrane, or even when the genetic material comes out of the capsid. Some theories say that the capsid is shed prior to entering the nucleus, but this paper proposes that the proteins on the surface of the capsid are essential to enter the nucleus through a nuclear pore channel. Nuclear pore channels already exist within the nuclear membrane to provide normal functioning for the nucleus. Now I'm going to give some background on the nuclear pore channel, or NPC, as it's noted throughout the paper. Here we have another piece of Figure 6, which illustrates how the authors think the capsid can go through the nuclear pore channel, which is on the bottom in gray and orange, and it allows it passage into the nucleus of the cell. So what are these nuclear pore complexes? Well, they're a type of entryway through the nuclear membrane that allows passage between the nucleus and the cytoplasm of the cell. They're necessary to translocate large species in and out of the nucleus because it needs some way to transport things across that membrane. There are different types of nuclear pore complexes, and they all have different entry requirements. This article specifically focuses on the phenylalanine glycine, or FG repeat domain types. It's important to note here that phenylalanine and glycine are both nonpolar amino acids, so FG repeats will have pockets of hydrophobicity. Previously, scientists thought that the central pore in the FG repeat nuclear pore channels was a maximum size of 40 nanometers. However, recent research from 2020 to 2022 suggests that this pore is actually 60 nanometers, which means it could potentially fit an HIV-1 capsid, which sits at about 60 nanometers in width, but it would be a tight squeeze. The FG repeat nuclear pore complexes are abundant, large, and they don't really have a lot of diversity in how they're made. They tend to allow cargo held within some sort of envelope through the pore, but not ever really a single molecule alone. Now, I'm sure you're all thinking, but Lainey, how can the HIV-1 capsid get through the nuclear pore complex if it sits at the maximum size? Well, the authors did address this, and their potential explanations of how the cargo cargo could get through include getting squeezed through the pore, simple enough. Charged species within the pore can bind to the cargo to pull it through, or even nonpolar molecules can bind to the FG domains, which are nonpolar, and enter through a concentration gradient. Or maybe, and this is their big one, the FG domains selectively bind to nonpolar targets, and the cargo will melt through the pore if it's recognized as valid cargo. Now we're going to go through the paper section by section and assess the experimental design and really see if the authors can back up their claims here. Each section in this paper is like its own little experiment, so we're just going to go through them one by one until we get through the paper. We're going to start with the first section after the background, which is called Important Independent NPC Targeting of HIV-1 Capsids, which is a mouthful, but let's get started and see what they were doing. The experimental design here was to check if they could see a few things. Number one, if they could catch an interaction between the membrane and their surrogate capsid sphere, and number two, if importans are necessary for the capsids to enter the cells. Importans are often used to help cargo pass through a nuclear pore channel. Here we have the first half of the experiment where the authors created a 40 nanometer sphere made out of the capsid protein on the outside and having a fluorescent label on the inside. The fluorescent label is necessary so that the authors can see exactly where the sphere is going. Then what they did was they incubated the free dye, the labeled capsid monomer, because each monomer was labeled here, and the 40 nanometer spheres independently with HeLa cells, which are a human cancer cell line. Then they measured the fluorescence to see how each species interacts with the cells. So let's look at the results. This is an image from figure 1, and what you're looking at on the left-hand panel is the free fluorescent dye. It's really important to use this as a control to see if your dye interacts with your experimental condition. As you can see here, the fluorescence is saturated throughout the image and really shows no localization, which is expected because it's telling us that there's no meaningful interactions between the fluorescent label and the cells, which is exactly what we expect. Now the middle panel is showing the labeled capsid monomer. In this, we see pretty much the same result as the free dye. There's no localization and therefore there's no meaningful interactions. So far, these results are exactly what we expect to see. The authors are showing us their controls, and their controls say that the free dye and the lone monomer are not interacting with the NPC membrane. Now, on the right-hand panel is really the actual experiment. This is the 40 nanometer sphere made out of the capsid protein, and it has the fluorescent dye on the inside. It's kind of a surrogate for our HIV-1 capsid, and that has undergone the same experiment as the other two panels where it's incubated with HeLa cells, and now you see that the 40 nanometer spheres are interacting with these membranes that contain NPCs. And we know that this isn't a coincidence because of the other controls. You can really see it on the GFP trace where you really see the outline of the cells from the fluorescent dye. And when you merge the NPC and the GFP trace, you can see that they're both localized to the same spot. My real critique here is I'm really wondering why we're not seeing any fluorescence past that membrane. The whole point of this article is to say that it's getting through the nuclear pore complex. I'm not really sure why we don't see any entry past that membrane here, but I think that the point the authors are trying to make is that they can measure what they're doing and that we can see an interaction with the membrane. They can catch it in the pictures. I think they really just took the picture quite early, and that's why we're not seeing anything on the inside of the membrane yet. Now we have the second half of this experiment to determine if the importans are necessary to bring the 40 nanometer spheres into the cells. On the left-hand side, they used a RAN-GTP mutated cell versus a non-mutated cell, which means that in the RAN-GTP, the importans will release cargo at the cytoplasm side of the membrane instead of on the nucleus side. What you can see is that the results really do look the same. The spheres are still making it to the nuclear pore complexes, so this really indicates that the important is not required. On the right-hand side, there's a corresponding control where the authors used an important beta binding sphere, and those were made to specifically require the importans to get through the nuclear pore complex. They used these spheres on cells with and without the RAN-GTP mutation, and what you see is that in the system without the mutation, the spheres look the same as our previous conditions where you see that there's some interaction with the NPCs. On the RAN-GTP mutation, you don't see anything co-localizing on the cell surface. It doesn't look the same as the control. And so the authors are saying that there's no real interaction here. My take, again, we're really only seeing this at the surface of the membrane. We're not waiting to see if there's fluorescent signal inside past that membrane. And in the important beta binding experiment, I see that there's diffuse fluorescence in the GFP and the merge trace, but it kind of looks like some of it got past the membrane. We see these little dots of fluorescence here, and the authors didn't really make a comment on that. So I'm not really sure what to say of it because the diffuse fluorescence tells me that there's no interaction. So it makes me wonder, maybe not all of them got the important beta binding label portion of it. Maybe there's something here that I'm missing, but I do think that they get their point across in their experimental design that they'd be able to successfully show the interaction of the capsid with the membrane at least. Now we're moving on to the next section of the article titled General Direct FG Repeat Interactions of Capsomeres. Now what the authors are looking at is the interaction between the HIV-1 capsid protein subunit, called a capsomere, and the FG repeat fragments within the nucleoporins, which they call NUPs. This experiment was designed using biolayer interferometry, which is a technique where you can directly measure how fast two species bind and release from each other. They found specific sequences in the nuclear pore channels that are rich in FG repeats and cut out those pieces. Where they cut out is noted after where it says NUP, and there's those two numbers. Those are the specific residues of the amino acid that they cut out, so you know exactly where they are. For more information on that, you can look in the supplemental. Those fragments that they're targeting were bound to a probe, and the probe was introduced to the capsomere, which is the subunit of the HIV-1 capsid. From there, they can measure how fast the capsomere binds to the NUP, which is what you're seeing from 0 to 200 seconds in all of the graphs. This is performed at varying concentrations, which is why you see multiple lines, and the results show that all of the FG repeat sequences shown here had strong binding to the capsomere. Then, they change the experimental conditions and allow the capsomere to dissociate at 200 seconds. That's why you see this decrease in response. These results show that the capsomere is able to bind successfully to the NUP and also dissociate. This is really important because it supports the hypothesis that the capsid will bind to the FG repeats to travel through, and it's not a permanent binding, it's just transient. Here we have the second half of the biolayer interferometry or BLI experiments. This is part B. What you're seeing is the important beta binding instead of capsomeres. So this is acting as the positive control to show that this experiment can be successful because we know that important beta will bind to the FG fragments in the NUPs already. And this data just shows that, yes, it's binding the way that it should bind. It may seem a little silly to include it, but positive and negative controls are really important, and I do like that this paper includes them throughout. Then in part C, we have binding with the normal capsomere, which is the colorful lines, versus the mutated, which is represented by the dashed line in each graph. So in this mutation, the FG motifs can't bind. In the middle chart for the NUP58, it looks like maybe there's a little bit of binding, but if you look at the scale, it's 0.5 versus 1.5 for the others, so we might be a little bit skewed there. I think we can reasonably say that if there is binding, it's very minimal. But the result from part C is telling us that there's specificity in these capsomere-NUP interactions where it's readily binding to the FG repeats and it's not something else. So my impressions of these results, again, we still aren't seeing the full capsid interaction yet. For this type of experiment, I think using the full proteins would be extremely challenging, and I honestly don't think that it would work anyways. I think there would be way too much going on and you wouldn't really be able to prove anything. I like that they use these targeted pieces to bind because it really hits home with where these interactions really are. But overall, I think it's good data. I think it does support their hypothesis, but it still doesn't prove it. This isn't enough for me to say, oh, this is definitely working, but they are on the right track. Moving on to the next section called Autonomous FG Phase Entry of HIV-1 Capsid Species. This experiment is exploring the entry of labeled capsid spheres into the cells again, but with a simple model. The authors created this model using FG domains based off of NUP98, which was tested during the BLI testing and also showed specificity for the labeled capsomeres. You may have noticed that from the previous experiments, they were using GFP as their fluorescent dye, and here they're using Sign GFP4A. This was done on purpose because the Sign GFP4A will repel the FG repeats and impede any potential binding, really just to make it harder for things to get into the cell. So what we're really looking at here is proof of concept data. On the left, you have the free dye, and as you can see, it doesn't seem to pass the membrane with the NUP98 in it. Then in the middle, we have the labeled capsomere, and you can see that it has some interaction with the outside of the membrane, but it doesn't go into the interior. The reason that this happens, the authors say, is because the fluorescent tag is exposed, and that's what's preventing the passage through the pore. Next up, we have our 40 nanometer capsid sphere back from those original experiments with the encapsulated fluorescent tag. And you can see that this obviously goes through the membrane. There's very clear localization within the membrane. You can see those beautiful green spots. So these results are great. They show that your experiment can work, but they really are just proof of concept at this point. Now we have part E from that same figure, which is evaluating different nucleoporins. They have the SLFG and FSFG, which refers to the different one-letter amino acid substitutions, which are different from the previous experiment, but they're still common domain types and they're still the nucleoporins. The result here is showing that the capsid can interact with a variety of FG repeat domains to enter the nucleus. And what we're really looking at is that localization of that green fluorescent tag. So you can see in each case, each of these three panels, you have localized fluorescence within the membrane, which means it's entering somehow. And we know that it's entering through the nucleoporins because that's the way the experiment was designed. It could only get through the nucleoporins. The authors also tested T. brucei all the way on the right, which has an FG repeat domain that's less hydrophobic than the others tested. This is notable because we're relying on this hydrophobicity to get through the pore to get inside the membrane. So seeing it work with a less hydrophobic pore is actually pretty cool. During this experimentation, the authors also note that the spheres pass through the pores quite fast, and my impressions here are really more of the same. It's good experimental design, but we're still not seeing it with the capsid. We're just still using those 40 nanometer spheres. Now moving on to the next section, complete NPC passage by capsid spheres. Here we're using the actual cell-based experiment instead of a surrogate. However, the authors are still using the 40 nanometer spheres and not the full capsid. What they did was they injected the 40 nanometer labeled spheres into the cytoplasm of a mouse oocyte cell and directly into the nucleus of that same cell type as their positive control. What we see in the cytoplasmic injection is that the fluorescence becomes localized inside the nucleus after 25 minutes, which shows that the capsid spheres are able to cross the nuclear membrane through those nuclear pore channels. In the nuclear injection, we see the fluorescence stays localized to the nucleus, and it looks the same as the cytoplasmic injection after the incubation time, which is exactly what we expected, so your positive control worked. My impressions for this section, I think all in all, it's a great experiment, but it really is just more proof of concept because you're still not seeing the actual HIV-1 capsid. I noticed that it's included as a really short section and the figure was only included as part of the extended data. I do like this experiment a lot, but it kind of makes me wonder why it's such a short portion of it. Now we're moving on to the next section of the paper, coincidence of FG binding and NPC passage defects. I think that this is really where we start getting into the meat of the paper. The first thing they show us is more of these FG phase experiments that we've already seen. And what we're looking at again is the 40 nanometer capsid spheres crossing a barrier rich in FG repeat domains. The experiment here is to compare the wild type or just the regular cells versus versus mutated cells. So we have that same N57A mutation that's been coming up throughout this paper. And as a quick reminder, this mutation is known to cause resistance, but not immunity to HIV infection. Comparing the wild type on the left to the mutated on the right, you see that the fluorescent tag makes it into the wild type cells, but we're really only seeing the surface interaction for the mutated cells. So this result is telling us that the 40 nanometer spheres can easily cross into the wild type cells. Mutated cells, they have a much harder time. Now what we have here is kind of a combination of the previous two experiments where we have the wild type cells versus the mutated cells. One of the most exciting experiments in this paper for me. And we have this mouse oocyte model again. What we're seeing here is that the GFP labeled spheres, when injected into the cytoplasm of the wild type cells, end up in the nucleus of those cells. You can tell that it's in the nucleus because you compare it to the positive control, which is directly to the right of it, and you see the pattern looks to be the same. So no problems getting into the wild type cells. Now moving on to the mutated N57A cells, in the cytoplasmic injection, it's really only making it to the outside of that nuclear membrane. Whereas in the nuclear injection, you see the dye in there. So what this is telling us is that it's really difficult for these 40 nanometer capsid spheres to get into the mutated cells. We had an idea of this from the previous experiment using those FG phases, which are man-made essentially. But here we're actually using cells. And I think that it's a lot more meaningful to use an actual cell than it is to use something that you create. But my impression here is that I really would like to see it with the full capsid. I know I say this after every experiment, but come on guys, we gotta get going there. Now we're in the last section, a cargo container for enclosed clients. This is maybe the biggest experiment in the paper to show us how the capsid could get through. Here's our design. This is more of the FG phase experiments, which is the model with a membrane with the FG repeat domain nuclear pore complexes, not an actual cell. And we have our negative control on the left of the free dye. This time they used M cherry, which is a red fluorescent dye, and it doesn't get past the membrane. You can see because in the M cherry trace and the merge trace, it's all black dots. Then we have the M cherry trapped inside the 40 nanometer spheres, or what the authors are calling CLPs, or capsid-like particles. These capsid-like particles are 60 to 80 nanometers in width by 100 to 180 nanometers in height, which is the closest model to the true HIV-1 capsid that we have in this paper. And that capsid, as a reminder, is 60 nanometers in width by 120 in length. So very close to our capsid-like particle size. We can see in this result that both the 40 nanometer spheres and the capsid-like particles are making it past the membrane through the FG repeat domains because we can see the localized fluorescence within those membranes. The authors note in the text that it looks like there's no size cutoff for these nuclear pore channels, but I mean, come on guys. Obviously there's an upper size limit if nothing else is the size of your nucleus, but I think there has to be some cutoff somewhere. I think they just haven't found it. It is interesting to see something that's potentially larger than the HIV-1 capsid making it through the membrane. I think these results are really great, and I think they're very compelling to see that this model capsid-like particle can get through their model nucleus. But I just really wish that they did the CLPs in the mouse oocyte cell experiment. I think that would really be a lot more compelling than what I'm seeing here. And I just don't really know why they didn't do that. They had the technology. They could have done it, but it just didn't make it. That's really what I want to see. And the paper here is just leaving me hanging because they didn't do it. Lastly, this paper wraps up with a diagram of the author's proposed mechanism of the HIV-1 capsid entry through the nuclear membrane. The capsid's outer surface, which is composed of the capsid proteins in blue, are going through the nuclear pore channel and melting in. So when they say it's melting in, they mean the FG repeat residues that are hydrophobic are binding to the hydrophobic outermost part of that capsid protein. And I think a really good way to think of this is that it's dissolving into the membrane, but it's only the outside that's dissolving into it. So the interior of the capsid is staying the same. It's able to deliver that genetic material into the nucleus, which is the whole goal of what it wants to do. I think that the mechanism is definitely convincing theoretically, especially considering all of their backup data, but I really would like to see a couple more experiments in actual cells with the HIV-1 capsid, and I think that there's definitely more to come from these authors. And now for the discussion. There's no beautiful data in the discussion. I'm sorry, the slide is very boring. The authors reiterate that they think the capsid is entering through the nuclear pore channels, and they refer back to their supporting data throughout. They also do acknowledge that their data was all in models and they didn't use actual HIV-1 capsids at all in this paper, which I think is really good. It's very important to understand your limitations and acknowledge them in your paper. And additionally, they propose explanations for why the capsid acts as its own nuclear transport receptor instead of relying on helper molecules. So they're saying that the single nuclear import event is enough for infection. Maybe they just don't need them, or maybe it's that the nuclear transport receptor coated capsid can bypass the size limits in the pore instead of having to have something help it get through. Either way, if it doesn't have to rely on anything, it's probably going to be easier to infect because the less interaction it has with the cell means fewer opportunities for the cell to be alerted that there's something wrong going on. So overall, my take, I think that this was a great paper, but I really am left wanting more. I think it's high impact, it's novel, but I really want to see more cell-based experiments from these authors. I think they really relied on these models very heavily, which is, it's the data that they have, but I would love to see some cell-based experiments. I also was really surprised that they didn't include computational data, so maybe that will be another paper upcoming. I would love to see a model with the actual HIV virus, like the full virus, including the envelope, starting outside the cell and getting through into the nucleus. I know it would be a monster experiment, but I would just love to see them tag how it moves to see what it does. Lastly, I think this title is very strongly worded for the data that they have. So just a reminder, the title is that HIV-1 capsids enter the FG phase of nuclear pores like a transport receptor. I think the data they have, they can't say for sure that that's how the HIV-1 capsids enter the FG phase, but it's a lot catchier than saying HIV-1 capsids might enter the cell this way. And I think that as a scientist, you see a lot of these catchy headlines and sometimes you don't feel like reading the whole article. So just keep in mind that when you write an article, you have to sell yourself. And this is definitely a strong title for the data that they have. And that's it for me. So thank you for watching. I hope you enjoyed it, and like and subscribe for more.